Hello everyone, and welcome to this School of Dentistry Research Showcase. Let me begin by acknowledging the traditional owners and their custodianship of the lands on which we meet today. And I pay my respects to the ancestors and their descendants who continue cultural and spiritual connections to country. We recognise their valuable contributions to Australian and global society. My name is Sasha Vanovsky. I'm a Director of Research here at the School of Dentistry, and on behalf of the Head of School, Professor Pauline Ford, and all our staff, it's my pleasure to introduce this research showcase. What we'll cover today is the main themes of the school's research capacity, and we'll also discuss our capabilities in the context of clinical research. A quick overview of the school. We are a relatively small school. We have 32 teaching and research staff, 7 full-time research postdoctoral fellows, and 25 HDR students. We are situated here at the Hurston campus in this magnificent, recently built oral health centre. And it's important to note that we have a very close alliance with Metro North Hospital and Health Services. So we're co-located here at the Oral Health Centre as part of the Oral Health Alliance. Some background to our research activities and some metrics just to show you the type of research we do and the quality and quantity of research we do. For a relatively small school, we are quite productive. Every year we produce somewhere between 250 and 300 publications, and most importantly, the quality of those publications is high. Approximately 20% of our publications are within the 10% most cited in their field. In terms of the type of research we do, it's also important to note that we are very transdisciplinary in our research. We collaborate and cross over many different fields of research, such as social sciences, engineering, cell biology and biochemistry, biomaterial science, and this will become more evident as we go through these presentations, and you'll get an appreciation of the breadth of research that we undertake at the school. We're also very good at collaborating. About 50% of our research is with international collaborators. And as you can see from this graph, that really lifts the profile of and quality of the publications. Those publications with international collaborators tend to be very highly cited and have a large impact. We also have approximately another 25% of our publications with national collaborators. So we like to collaborate, we're open to collaborations, and we very much encourage collaboration. Our facilities here at the Oral Health Centre, we're very fortunate to have quite a substantial PC2 lab with OGTR certification, and we have also clinical facilities which we'll touch on more in due course during this presentation. In terms of the PC2 lab, we do have a number of facilities which are unique to the HABS faculty, and in some cases the university. So in some cases common things like microbiology and cell culture. In other cases quite unique capacities such as hard tissue histology and biofabrication facilities. We do have research embedded in our undergraduate program, so all of our students finish with an honours. Our program is an honours program, so all students undertake research during their program, and you can certainly get an appreciation of this research in these publications of the abstracts and the research from each of every year's undergraduate research conference, and this can be found in the library and online. The research themes within the school, we have 3 main themes: advanced materials and technologies, which is led by Professor Laurie Walsh; dental public health, currently led by Associate Professor Rati Lalu; and tissue engineering and additive manufacturing, led by myself. It's also important to note that we are a clinical school, so we have substantial clinical research capacity and expertise, and we have this expertise organized according to different research, different disciplines, clinical disciplines within the school, and we'll touch on this more as we go along. But now I would like to invite Professor Walsh to talk about one of the main themes of the school's research, which is advanced materials and technologies.
Thanks very much, Sasha, and it's good to be able to talk with everyone else today. Our research strengths in this group range from nanotechnology through to photonics and imaging, and obviously dental materials science, which includes some of the more chemically based materials and also things which have got greater biological impact. And so we do interface very much with clinical research. We work a lot with complex biofilms, and we also are interested in how to translate what we're doing back to the clinical environment. One of the unique features of our group is we have roughly 90% of the university's fleet of medical lasers live in the School of Dentistry, and that's been very useful for us doing some of our research in advanced imaging and in photonics. So within UQ, we're very much connected to IMB, engineering, SCMB, PACE, ARBN, and most recently also to the vet school. And we collaborate intensively with universities in Australia. Particularly, we're linked with health services in Metro North and also at Mater and also with Metro South. And we currently have 2 industry programs, one based here in Brisbane, the other one based with some collaborators in Canberra. In terms of outputs, this group's been reasonably productive. When I ran some numbers just over the last 3 years, we publish on average at a reasonably good rate. And recently we've just completed a book, which is mostly dominated, not surprisingly, by people from the school. Who would have thought? Many of our publications are not in dental journals. You can see that we publish in physics, engineering and materials journals, and also in oncology journals. And we've had a fairly good output of research students, and we've got a good number who are currently enrolled with us. So what do? Well, in the nano world, we work with nanomaterials, nanoparticles, nanofibers, and also protein and mineral nanocomplexes. And we've done that both in the laboratory, but also in clinical research. Our research includes research into light-sensitive nanoparticles for gene delivery. And Chun Zhu, who's shown on the slide here, who's listening to us today in this presentation, Chun has led a lot of the nanoparticle research. We've tested nanoparticles that can alter complex biofilms. And we've more recently got into working with nanofibers, and particularly nanocellulose. In the photonics and imaging area, we've done work with laser scanning, with the interpretation of conventional radiography, with using lasers in different ways, for example, to do detection of different types of biofilms and bacteria, and also how we can deliver the laser energy better. In the area of dental implants, what you can see here are the surface of some different sorts of dental implants. We've looked at the problems of trying to clean these surfaces and how we can deal with those in new and better ways, and even the technology of placing and servicing an implant. And obviously this involves our team members who are specialists. In this case, the specialist prosthodontists have had a fairly big input into that. In the area of endodontics, our school is very strong, having recently recruited Professor Ove Peters to join a very strong team of researchers in the school, working across the range of things from vital pulp therapy, in other words, keeping a tooth alive, through to new sorts of materials, new sorts of instrumentation, and even the way the instrumentation is actually used. And then finally, in the clinical research area, we've had a very strong paediatric dental caries program that's been doing a very, very long-scale clinical study, one of the largest of its type actually in the world. We have another program that's been looking at human papillomavirus in saliva, and we link very strongly to one of the clinical research units that Sasha mentioned earlier, which is looking at issues around care in hospitals and also care in residential aged care facilities. I'm going to now pass on to Rati Lalu, who's going to talk you through dental public health.
Thanks, Laurie, and hello to everybody. I'm going to talk to you about dental public health. I'm the discipline lead in this research stream. And dental public health is a fairly broad area of research, but essentially we're looking at the epidemiology of health and oral ill health, but also importantly looking at opportunities to improve and enhance the oral health of communities at the population level. So I'm just, the team, fairly broad. And this is probably the most important team. We have the head of school in our team. So we'll say we're the best. But there's various other folk in the team, and you can see them listed there. And we've also got numerous collaborations across the university with external universities and institutes, but also importantly with the local hospital and health services, particularly Metro North, West Moreton, but also Metro South. Now, just a quick snapshot of the global burden of oral conditions. More than half of the world's population will suffer oral diseases at some point of their lives. And in fact, caries of the permanent teeth is probably the most prevalent condition globally, with almost 2.5 billion people suffering caries of the permanent teeth, and almost 500,000 people, 500 million people suffering caries of the primary teeth. Now, I particularly highlight that because of my contribution within the Global Burden of Disease study, which is, I assume many of you are familiar with. It's a significant undertaking to give one a general picture of the global burden of numerous diseases, including oral health. And what's happened since a number of these publications is, in fact, the WHO is encouraging member countries to now recognize the priority of oral health and strategize in terms of reducing the burden of those diseases within countries. Now, that might sound like a trivial thing, but it's actually quite complicated to get WHO to make note of this condition. I've also been fortunate to work within the injury space, and the bottom paper, which is a paper I led, and there are opportunities within this group to actually access the data and write publications in areas of interest to you. So I'd encourage people to look at the global burden of disease, and particularly the visualization of the data. Another significant piece of work, and a lot of our work tends to obviously focus on Australia. And again, just another quick snapshot there. For example, about a quarter of adults have untreated tooth decay. We still don't have water fluoridation across all parts of Australia, particularly in Queensland. And there's still issues around sugar consumption, smoking, and risky drinking. So some of my work that I've been involved in particularly is in North Queensland, remote North Queensland. We were quite fortunate to have a fairly significant NHMRC project grant to do an intervention around caries burden. As you're all aware, particularly in Indigenous rural communities, the burden of oral health is particularly massive, particularly compared to non-Indigenous counterparts. So it is absolutely crucial we do trial various interventions in these communities. And the study went on for about 4 or 5 years. And essentially the intervention that we've trialed clearly shows that it was effective, but also cost effective in reducing the burden of dental caries. What was also an added bonus of the study is, in fact, the funding was also provided for treating the dental caries in this community. So in fact, all the kids that participated were made dentally fit. All the caries was treated before the prevention was applied. So those are just some more publications. And again, the important part here is the translation. And we've now got a new state health minister, so hopefully we can progress this in terms of translating and making sure that the burden of caries, particularly in these communities, is addressed. Just some pics from parts of the world in northern Queensland. We were quite fortunate at the time we were doing the study. The cabinet, the federal cabinet, actually met in northern Queensland, and you'll see the then federal minister of health talking to us. But also that the sign is the most northern point of continental Australia. So it's a wonderful opportunity, absolutely beautiful part of the world. There's also been a recent announcement that we've recruited Professor Logdo from the University of Adelaide. Logdo comes with enormous experience in dental public health, and he will come along with significant grants, including this one, which is going to look at the impact of water fluoridation now that it's been fairly widespread in Queensland for about 10 years. So that's a significant study over the next 4 years and includes Pauline Ford and myself from the school, but various other folk within Queensland also nationally. So that's going to be a significant piece of work that's being planned for the next few years. He also has a number of other projects which he will come along with, and the Smile Birth Cohort study and the Tooth for Health study. These are largely based in South Australia, but he will carry on this work when he comes to UQ. Now it gives me great pleasure introducing Nicole Storman to talk about other aspects of our work.
Thank you. So under the supervision of Professors Lalu and Ford, my research has modelled children's oral health using the longitudinal study of Australian children. And a key collaboration that was developed throughout my thesis was with the Murdoch Children's Research Institute. And so through the Child Health Checkpoint study, which was a nested study within the LSAC, a subset of the birth cohort had dental photographs taken, as you can see on the screen, as well as 3D composite images of the face, which I analysed. Most significantly, this data will form parts of future releases of the public domain of the LSAC data. So I analysed the broad epidemiological aspects of the data, and it will be linked to this long and a large cohort study. So additionally, in collaboration with Metro North, we've conducted a series of studies assessing the effectiveness of a streamlined and facilitated pathway to dental services for people experiencing homelessness. This project primarily identified that this population experiences fear and shame as a major barrier in accessing dental services. And our research has helped identify a cost-effective and sustainable service model for this population. Another area of research within our group is on oral health and cognition. As you can see on the screen, Drs. Nangal, Manchuri and Pradhan have a series of longitudinal studies that they've looked at, looking at cognitive predictors of oral health in older adults. As part of Nathan's thesis, they have also conducted a body of work with people living with multiple sclerosis. So as you can see from our slides and also our publications on the screen, our research in dental public health covers a number of topics, populations, and collaborates with many universities and organisations. Our publications and research areas can be found on our research profiles online and also on the School of Dentistry's website. So thank you, and I'll pass you back over to Professor Ivanovsky.
Thank you, Nicole. I'd like to now spend a few minutes to talk about the tissue engineering and additive manufacturing theme. The first thing I'd like to say about this theme is that it's recently transitioned into the Centre for Orofacial Regeneration, Reconstruction and Rehabilitation. And part of the reason for this is because the group has been quite successful and quite productive, and we wanted to allow some of the very talented mid and early career researchers to develop their own leadership skills and establish their groups within the centre. So Dr. Cedric Vaquette, Dr. Karan Gulati, Dr. Ping Ping Han and Dr. Abdallah Ali all have the opportunity to now start to establish themselves as independent researchers. And you'll hear from all of them in the next few minutes. I'd just like to talk to you a little bit about the group as a whole. The overall focus of the group is on novel approaches on solving common dental diseases, especially focused around bone defects and various structures that support teeth, which are the periodontal structures that are usually affected by these very common conditions, which are periodontal disease, one of the most common diseases that affects humanity. Within the program, we have 4 academic staff and 6 full-time research postdoctoral fellows and 13 HDR students, 12 PhDs. Over the last few years, we've published extensively, and these papers have been also very highly cited, and we've had significant success in various competitive research grant applications. The crux of our approach is to utilize 4 IR technologies. So this is technologies that are focused on digital data and digital technologies such as imaging. So the idea in applying our approaches would be to utilize advanced imaging techniques and advanced additive manufacturing, such as 3D printing and electrospinning, to then combine this with know-how and expertise in clinical regenerative medicine and dentistry, and ultimately to be able to produce personalized or custom constructs for patients. The nature of periodontal defects and other defects within the mouth is that they're quite specialized and quite specific in the way that they present. So a customized approach is very suited in these cases. We have a variety of additive manufacturing capacities, and we'll discuss this more during the remainder of these presentations. But as a summary, we have 4 different printers. They're both, most of them are capable of bioprinting, which means viable constructs for implantation in vivo, as well as melt electrowriting technologies, which are quite unique within the university. So this is an example of one of our bioprinters producing an implantable customized scaffold. This has been designed from a digital file of a specific periodontal defect, so we are able to produce this highly porous construct that is able to then be inserted into patients to achieve regeneration. We also have quite a significant capacity in terms of histology, and in particular, something that's unique again within the university is this ability to process non-decalcified tissues using our hard tissue sectioning and processing equipment. This is also suitable for histology of metal implants and other hard tissues without the need for decalcification. We have a whole suite of mechanical testing materials and equipment which are used to characterize our suite of biomaterials. A very important and significant recent development has been our collaboration with the Hurston Biofabrication Institute. This is a recently established institute by Metro North at the Hurston campus. It is certainly a very significant undertaking. UQ is the academic partner in this institute through a collaboration of several faculties and also the AIBN. And the School of Dentistry has just been announced as a partner in the latest program, which is the craniofacial program that's being funded by the Institute, and the funding for this is $4 million over 5 years. So this is certainly a very significant collaboration for the school and for this particular group, because the expertise and aims of our centre and this institute are very, very closely aligned. So now I'd like to invite Dr. Cedric Vaquette to talk about his recently established group within the centre.
Good afternoon, everybody. As Professor Wienowski mentioned, my name is Cedric Vaquette, and I'm leading the Biomanufacturing and Regenerative Dentistry group. And our group is actually interested into 2 main themes. The first one is obviously regenerative dentistry, being in the School of Dentistry, where we actually do a lot of work around the periodontal regeneration, extraskeletal bone augmentation, and also soft tissue expansion and grafting. And the other topic, and it's closely linked with the techniques that we use for the first theme, is actually regenerative dentistry and for application in orthopedic. So we do a lot of work around ligament and tendon, bone regeneration, and we're also quite interested into investigating how the biomaterial architecture that we produce influence the mechanism of bone formation. So our research strengths comes from the fact that we are actually a pluridisciplinary team. So we are composed of biomaterialists, such as myself, but also biologists and clinicians. And we are really focused onto clinical translation, which means that whatever we start developing is designed with the end goal of being able to place those constructs into a patient. And this is particularly important for what we do, and that's why we have actually developed a number of quality management systems around our manufacturing production. And another strength that we have is because we are using additive manufacturing, we are actually developing a highly complex structure that could be a multi-phase structure that could be used for highly hierarchical organ regeneration. So as mentioned before, our expertise mostly relies on additive manufacturing technology. So we do a lot of 3D printing, we do a lot of bioprinting, metal electrowriting, and of course, we have also the capacity of testing those biomaterials, so both biologically, but also their physical properties. And then one step further, we have actually a lot of expertise around the development of preclinical animal models to assess the regenerative performance of those constructs we are fabricating in our lab. And another expertise that we have is the convergence of all those additive manufacturing technologies. So the facilities that we have were just presented by the previous speaker, but to summarize this, we have an additive manufacturing lab with various 3D printer, a mechanical testing lab, and we have the capacity of analyzing the tissue that we retrieve from the animals. And we have also a cell culture facility. So just to highlight some of the type of research that we do, so we are quite interested into periodontal regeneration. So the periodontum is actually all the supporting structure that provide biomechanical stability to your tooth. So this is composed of bone, a soft tissue, which is the periodontal ligament, and then a cementum, hard tissue cementum, which is laid on the root surface. So all of that contributes to ensuring your tooth stays where it is. However, that organ can actually be attacked by a disease called periodontitis, which is caused by the infiltration of bacteria. And those bacteria and the formation of biofilm creates an inflammation, which in turn start destroying the bone, and you lose attachment. So of course, you may think this is just a tooth, but actually it is quite important to address and to stop the progression of this disease and to regenerate the periodontum, because actually patients with periodontitis have been associated with many other systematic diseases. So basically it seems that periodontitis might be the gate towards development of other more debilitating and potentially life-threatening diseases. So that's why we think this area of research is very, very important. So to try to regenerate this tissue, we have actually developed a number of techniques, and all of those techniques are revolving around tissue engineering principles. So we have used cell chip technology, we have used decellularized matrices, and the cornerstone of this research program is really the development of biphasic 3D construct, 3D scaffolds, with fiber guiding capacity. And there is an example of such a construct that you can see on the top row of that image on the right, which is manufactured by metal electrospinning writing and has fiber guiding capacity. We are also interested in developing in vitro models for periodontal disease. And again, the technology we use here is actually metal electrospinning writing for developing those fiber guiding scaffolds and bioprinting. Another aspect of our research is investigating the regeneration of bone in the extraskeletal area. And this addresses a clinical need. Basically, when a dentist wants to place an implant, sometimes it can't because there is not enough bone. So he has to grow bone vertically outside the bone envelope before he can place the implant. And this is relatively successful. However, when the implant is placed, there is usually a lot of resorption of the previously formed bone. And what we have investigated here is actually whether the utilization of porous and biodegradable scaffolds can prevent this resorption. And we were successful in showing that actually it does remain. The bone does remain post-implant placement. Another aspect of our research is more on orthopedic application, where we look at ligament and tendon regeneration. And usually this is achieved by the development of bone, ligament, bone multiphasic construct. And some of that research has been actually recently published. And last year we were lucky enough to be highlighted in Advanced Science News for this work. So to assess the performance of our biomaterials, we have actually developed a number of animal models, starting from small animals such as rats, where we can implant subcutaneously, or we can place in a bony defect, sheep, and also pigs. So if you have a construct that you would like to implant in an animal model, please come and talk to us, because we have a lot of expertise in this area. So one of the last slides is actually the different type of funding that we have, and also just to show as well that we are collaborating at the local level, but also international level. So to summarize our group, we are a group that is focused on addressing clinical needs in dentistry and orthopedic. Our expertise rely mostly on additive manufacturing and the development of preclinical models, animal models. And we are really keen on collaborating with people that have knowledge on bone and soft tissue biology, drug delivery, and also the development of new hydrogel for bioprinting. And our group is actually one of the projects of the group is actually trying to create bone to place implants, and the next speaker will actually enlighten you on how you can actually increase the stability of those implants.
Thanks, Cedric. Hello, everyone. My name is Karan Gulati, and I lead a group focused on anodized therapies for osseointegration, regeneration, and stimulation. So basically, we perform the nanoengineering of metal implants to fabricate these beautiful nanostructures so that the bioactivity and the therapeutic potential of conventional implants can be enhanced. Our research is focused on different areas. First is optimized nanoengineering, which involves fabrication of controlled nanotopographies on various metals, alloys, and commercial implants to ensure clinical translation. Another focus is the local drug release. So in theory, we can load any drug, protein, or growth factor inside the nanostructures, which will then be eluted locally once the implant is placed. The focus is on tailored bioactivity and therapy, that is enhancing the soft tissue integration, osseointegration, immunomodulation, and achieving effective antibacterial efficacy. So we fabricate controlled nanotopographies, and you can see on the slide here, we can control the topography, the chemistry, and the drug release of it. The focus is dental implant challenges, although the technology translates well into other implants like orthopedics. So we start with electrochemical anodization, which is a very simple, cost-effective, and scalable procedure. What we do is we take the conventional implant and we immerse in an electrolyte which has fluoride, ions, and water under optimized conditions. When we flow appropriate current and voltage, such nanostructures will self-assemble onto the entire surface of the implant. Now here you see the nanopores, which are like tiny test tubes, open at top and closed at bottom, forming all over the surface area of the implant. We can modify any implant surface, even the ones which have micro roughness. And we can translate the technology into implants of different sizes, geometry, shapes. And because they are empty test tube-like structures, they can be loaded with proteins, growth factors, or antibiotics, which will then be released locally once it is placed inside. Because they are nanostructures, nanoscale in roughness, they promote the activity of the different cells. So we can control the bioactivity of various cells here. Here you see some of the nanostructures that we have optimized in last couple of years. So we can fabricate the good-looking nanotubes. You can see they're perfectly cylindrical, open at top and closed at bottom. More recently, we transferred the technology into fabricating more mechanically robust structures. And on the bottom, you see here are aligned nanopores, nanotemplate, and random nanopores. Now, based on the alignment of these structures, the dimensions of these structures, you can control how the cells will behave. This will also control how much drug can be accommodated and accordingly the release kinetics. Like I said, we can preserve the underlying micro roughness, which is very important in dentistry because most of the implants are already micro rough in nature. So our technology preserves the micro roughness and on top of that superimposes the nanotopography, which has not been done before using this technique, which is electrochemical anodization. As I've told you, we can fabricate and control the alignment of the nanopores. You can see they're aligned, they're random, and also we can fabricate nanotemplate. We can also fabricate nanotubes if we want more drug to be loaded inside the nanostructures. The bioactivity can be controlled depending upon the characteristics of the nanostructures. Here I just demonstrate how we can control the activity of 3 different cells. On the left-hand side, you can see the primary macrophage morphology is more circular in nature, which indicates a more inactivated state, whereas the osteoblast and fibroblast activity is preserved and enhanced, and the cells tend to align parallel to the nanopores, which is a strong indication of mechanostimulation. Here we see the titanium nanopores, TNP, at different diameters, and you can see how the cells are happy and the cells are aligning almost perfectly parallel to the underlying nanopores. Again, strong mechanotransduction here. We have been fortunate to have collaborated with leading research groups all over the world, and also thanking the funding bodies here, especially the NHMRC, JSPS, and ADRF. Our technology has also featured in different media platforms, especially the part where we are talking about improving the success of dental implants by incorporating antibiotics, growth factors, and other therapeutics. So what are the strengths of the group? So we are expert in electrochemical nanoengineering, so we can control the topography and chemistry of the various nanostructures. We can fabricate these nanostructures on different implant substrates. We can even preserve the micro roughness, that is, you get dual micro and nano benefits of both the worlds. In theory, we can load any drug, protein, or growth factor inside the nanostructures, and its release can be tailored to match specific therapeutic window. We are looking at different research projects or ideas to collaborate with other researchers, including infection models and quantification of release inside the tissue ex vivo or in vivo. We're also looking at modeling, the drug release modeling, advanced cell stimulation, and also quantifying the cell cytotoxicity. And now I would like to welcome Dr. Pingping Han, who will be talking about her group on epigenetics and nanodiagnostics.
So our group is focusing on the epigenetics, especially in diagnosis and therapeutics. So in our group, we have 3 main themes. One is the exosome or extracellular vesicle as biomarkers for oral disease. And the second theme is exosome or extracellular vesicle as a therapy for periodontal regeneration. So as you know, you may not know, so the exosome or EVs, they are basically, you can imagine that they are the body of your cells, and they are floating around in your saliva and GCF and blood in the body fluid. So we aim to use these small nanoparticles can be for the biomarkers. So in our lab, in our group, we have optimized the isolation from both saliva and GCF. And further research will be done in other biofluids like blood and some other urines and stuff. Also, our group already have done some research on the periodontal regeneration, as Dr. Cedric already said about this part. And here is the 2 of the publications we have done over the past few years. So based on this foundation, we are going to investigate whether this exosome can be another regenerative medicine for this disease. The third theme is to investigate the nanoscaled mechanics in the cell, how they respond on the different modified substrates. So based on what we can see here, that first you can see how your molecular protein are located into your cells, and also we can use the same biosensor to use the biosensors to detect the nano piconewtons on the cell surface, on cell edge, which can direct the cell fate in the end. And our expertise lies on different fields. One is the exosome biology and also the molecular biology. So we are the one of the labs in the world which can isolate the exosome or extracellular vesicles using SCC method. SCC method is one of the most modern technique in the world which can isolate pure population of the EVs. And to be noted that we have the in-house SCC columns which can significantly decrease your research cost compared to the commercial ones. And then you get a pure population in the end, and then you can utilize those purified EV populations into the animal work and even in the clinical trials in the future. And also we have the ChemiDoc, which can be used for Western blot and some other fancy techniques. The third and the only one, the equipment I want to mention is the Lonza nuclear factor, which is the only available equipment in Hurston campus. This small equipment is very powerful, which can only allow these hard to transfect cells to be transfected with different biosensors, like I mentioned in the last slides. And anyone who wants to investigate more biosensor work and mechanical transduction, you can reach us using this equipment, and we can collaborate. And also our people are included on masters and visiting scholars and also declinant students. And our research was significantly collaborating with different collaborators within UQ in CCR, AIB, and QBI, and also some other units in Australia and worldwide. And our fundings are from different funding bodies as well, including UQ and some other societies. And this is one of our research were featured on the news that saliva can be a diagnosis test for the COVID-19. And next, I would like to introduce our colleague Ali.
Thanks to my colleague, Dr. Bingbing Han. Hello, everyone. My name is Ali Abdullah. I am a group leader of biodegradable and biofunctional biomaterials, simply A3B. So our research team designed advanced biomaterials and multifunctional characteristic for soft and bone tissue regenerations. Our expertise about additive manufacturing technology using a 3D printer, metal writing, solution electrospinning, and freeze drying. Biomaterials characteristics include not limiting to physical, chemicals, mechanical, and biological analysis. Biodegradable metal implants, especially we are focusing on magnesium-based alloy, zinc, and iron-based alloy as well, as we have a broad expertise on surface modification of magnesiums and how we can control its corrosion properties. And recently we are focusing on zinc-based alloy as transferable, transportable materials, biomaterials for bone tissue augmentation, as I will explain in a minute. Biodegradable polymers and nanocomposite scaffolds. So research facilities including mechanical analysis, tensile, compression, fatigue, micro and nano indentations, chemicals, FTIR analysis, thermal properties, XRD, XPS, and cell culture core facilities are available. 3D printer using Envision Tech, bioscaffolder, metal writing, and freeze drying, as I mentioned. As you can see from the shown image about using 3D titanium mesh for bone tissue augmentations, but actually titanium implants, it's need for a second surgery operation. So our focus, how we can really use a 3D biodegradable scaffold for vertical bone augmentations, including zinc-based alloys, magnesiums, and compare these properties with BCL custom mesh implant. So our people visiting scholars from Chumbak National University and Dr. Ayush and Mr. Ayush and Sina. We have a strong collaboration nationally and internationally. Many of them had got really many numbers of publications in high data and well-known journals. We currently collaborating with pioneer professors from Germany, from MIT and Harvard universities, and from as well Dunhua University. So our work has been well recognized in social medias recently. Really our story has been published in Create, Australian Engineers Australia, and also Korean Mail's Australian newspaper. Why you should collaborate with us? So we have a strong track record in the field. We are capable of publication in high-ranked journals, exchange ideas, strengthening our profile together. We are productive. We used to publish from 7 to 10 papers in high, well-ranked journals every year, and building a strong collaboration for attracting grant funding, clinical transaction, and commercialization. So next, I would like to introduce Dr. Lee, talk about UQ clinical research units.
Thank you, Ali. My name is Ryan, and then I am the lead for the clinical research unit. As a clinician at the same time being in a very clinical school, our main aim is trying to facilitate the clinical and translational research, all the great developments and then in dentistry behind the scene, all the clinical studies we're trying to implement for the care of the patients who are suffering from oral and dental diseases. That includes the research, the public dentistry as well. So our expertise lies in many disciplines of dentistry, starting from radiology to all the way down to the special needs dentistry. And then being a dental school and one of the leading dental schools in Australia. And then we are putting a lot of emphasis on the clinical research units. And also we are collaborating, we create a good alliance between the UQ and Metro North, and that gives us a really good position to have access to all the public patients, hence we can conduct those researches in the future. So our clinical research unit within this lies within this great building. Oral Health Center is located in Hurston, and the clinic for the facility for research unit will be located in level 6. And we have 4 modern dental chairs where we can conduct many different type of dental procedures, starting from the exam to all the very complex procedures. And then we also have an additional room for patients' interview, meeting rooms, any type of discussions. Of course, we have a reception, waiting room. As a normal dental clinic, we have all that. In addition to those normal dental facility, we have a very great access. We have a great access to the PC2 lab. So whatever we do whatever we collect from the patients, it can be analyzed straight away. In addition to this great facility, we also have some agreements with the private practices around the Brisbane area, and then obviously they will be deemed as satellite clinics, and we can widen our patient pools, not just looking at the public patients, but also including private patients. Hence, it can be more applicable to overall general publics. And our people involved in this particular clinical research unit, again, we have Professor Walsh and Professor Peters. We have all these people involved in this particular clinical research unit according to their disciplines. So what are the research themes for clinical research unit? Well, it's basically collaborating between the discipline. At the same time, all the talk we just had from our colleagues about the great developments of the dental research, we want to put them into putting into the translational dental research so we can actually benefit the patients. For that, we are also having a lot of collaboration from international institutes, including UK ones, Spain, Europe, throughout the Asia, and also in America. We have various funding sources. Being a dental school, we have a lot of help from the dental companies, including Colgate, and also the implant companies and material companies is helping us quite a lot. And then we also have a great success in NHMRC. And then as an industry, the Australian Dental Research Foundation, we get a lot of funding from those particular funding bodies. To highlight some of the projects going on in our clinical research unit, well, we have a lot of randomized clinical trials looking at periodontal unit. This is just example of the periodontal clinical research unit. Prof. Lalu and Nicole has already gone through what's going on in the public oral health sector. And then Prof. Walsh also touched on his research on the pediatric dentistry. And this is just example of the perio unit. We are looking at many different aspects of the periodontal disease, how we can improve the treatment quality and also the prognosis of those, the basically treatment outcomes of those newly developed treatment strategies. We are also implementing, and we're trying to see how we can improve the bone preparation for the implant treatment as well as an implant research unit. And prospective clinical trials also going on, looking at the popular topic of vaping also, and then see whether we can improve the periodontal attachment benefit by using different materials, hence patient can avoid future problems. Recently, we've been collaborating with the Colgate, looking at phase 3, the clinical trial, looking at new particular toothpaste, and then obviously that will be expected to commence in 2021.